All right, so this is the last video for the respiratory system chapter, and in this video we are going to talk about external and internal respiration, which is just gas exchange either in the lungs, between the capillaries in the lungs and the alveoli, or in the tissues, which is internal respiration, so between the capillaries and the tissue cells. And then we'll also talk about how CO2 and oxygen are transported in the blood. All right. And then at the very end, we'll just do a little discussion on how respiration is controlled. Okay, so let's start with external respiration. Before we get into kind of the details of external respiration, we have another law to discuss pertaining to gases, and this one is Dalton's law. Okay, and so Dalton's law just simply states that the total pressure of a mixture of gases is equal to the sum of each of its individual gases, the pressure for each of those individual gases. All right, so we refer to that as the partial pressure. So, for example, the air we breathe, right, our atmospheric air has multiple gases in it, including carbon dioxide and oxygen. All right, so oxygen would have its own partial pressure. So we just mark that as PO2. All right, that's the partial pressure of oxygen. That's just the pressure for that specific component gas. And so then the partial pressure of carbon dioxide would be its own separate pressure. Okay? All right, so let's talk about external respiration. All right, with external respiration, this is gas exchange between the alveoli and the capillaries. All right, so this is occurring in the lungs. Okay, and so with external respiration, it's going to be important to keep in mind that the partial pressure of oxygen in the alveoli is essentially constant. All right, and that's because the alveoli have a constant supply of oxygen from the atmosphere. Okay, so that partial pressure in the alveoli, when we look at our picture, is not going to change. Okay, there are factors that can affect how efficient external respiration is. So let's go through those really quick. So the first factor would be diffusion distance. All right, so this would be related to the thickness of that respiratory membrane. All right, so this is just the distance that those gases have to diffuse across in order to move from the capillaries to the alveoli or the other way. Okay, so the diffusion distance. So this could be changed by a number of factors. For example, fibrosis of the lungs. If you start adding a lot of collagen fibers, all right, between the alveoli and the capillaries, that's going to increase the diffusion distance, as well as edema, right? Edema or even pneumonia, where you get fluid building up in the lungs. That fluid will actually increase the diffusion distance and make it more difficult for those gases to be exchanged. All right, so that's one factor. Another one is ensuring that there is adequate ventilation. So ventilation just refers to airflow. All right, so adequate ventilation, there's nothing preventing air from getting to the alveoli. There's no obstructions or anything like that. Okay, so this could be altered, for example, with something like COPD or asthma, right? So obstructive diseases. Right? The lungs also need adequate perfusion. That would be blood flow. All right, so obviously important because the two things where gases are being exchanged would be between the capillaries, which is the blood, and the alveoli. So you need enough air getting to the alveoli, and you need enough blood getting to those capillaries. All right, so this could be related to the diameter of our arterioles, if they constrict and prevent blood from moving in. The blood, if there's an increase in viscosity of the blood, that could slow down perfusion. Anemia could decrease perfusion. just decreasing that oxygen carrying capacity, or even something blocking, right, like a clot or a thrombus blocking blood flow into the lungs. All right, pulmonary embolism could do that also. Okay, so we need good blood flow, good airflow. And finally, the alveolar diffusion gradient. All right, we need a diffusion gradient. And so this means that gases are going to move, right, like we've said before, from high to low pressure. So in order for oxygen to move from your alveoli into the blood, the partial pressure of oxygen in the alveoli has to be higher than the blood, all right, for it to have a pressure gradient to move down. And so then we also need to have the carbon dioxide in the blood be higher than in the alveoli in order for that CO2 to be moved into the alveoli to be exhaled. Okay, so we do need a diffusion gradient, a pressure gradient for the different gases we want to exchange. Okay, so those are important for external respiration, that's in the lungs. And then there's also internal respiration, and then we'll kind of look at putting these two together and the different pressures involved. All right, so with internal respiration, this is going to be between the capillaries and the tissues. All right, and so we would just invert things, okay? So for internal respiration, our oxygen would be higher in the blood because it's oxygenated blood than in the tissues, and we want that oxygen to move from the blood to the tissues. And then CO2 in the tissues would be higher than in the blood. Okay, so let's look at some actual pressures and some examples of this, all right, with this picture. And so what we're looking at here, just label a couple things. This would be an alveolus. All right, so we would have external respiration would be up here. All right, and so then we have the blood, after it picks up its oxygen, becomes oxygenated, travels through the left side of the heart and out through the aorta to the tissues. And then down here, so we would have the tissues here. All right, this would be internal respiration. All right, and then that blood would continue back through the right side of the heart and go through external respiration again to pick up oxygen and get rid of carbon dioxide. Okay, so let's talk about the gases. So in the alveoli, we said that the partial pressure of oxygen is relatively constant. That's because it has that source of oxygen from the atmosphere. And so it's constant at about 105 millimeters of mercury. All right, so that's typical for that atmospheric air. All right, and then typically carbon dioxide, all right, in the air we breathe would be about 40 millimeters mercury. Okay, so blood coming from the body that's deoxygenated, it's coming to the lungs to pick up oxygen. Remember, we need a lower PO2 than what we're getting in atmospheric air. So this deoxygenated blood has a partial pressure of oxygen that is about 40 millimeters of mercury. So definitely lower than that 105. All right, and so what's going to happen is this oxygen, all right, is going to move from high to low pressure. All right, so the O2 is going to move into the capillary, all right, until that capillary reaches a PO2 of 105. Whoops. Because remember, that stays pretty constant, so it's not decreasing as that oxygen is moving in, okay? All right, so the partial pressure, so oxygen moves from its high pressure to low pressure, it's going to move into the blood vessel, and then that PCO2 is going to be higher in our deoxygenated blood, not too much higher, it's about 45 millimeters of mercury on average, okay? And so it's going to want to move out, all right, from high to low pressure. All right, so it moves out to the alveoli where it can then be exhaled, all right, in the air we breathe out. And so that would leave the PCO2 in the blood at 40 millimeters of mercury. Okay, so when the blood passes by, right, this alveolus, essentially it takes on the properties, the partial pressures that were present in that atmospheric air. Okay, so it exchanges, gets rid of that CO2, and it picks up more oxygen, so that now the partial pressures in the blood resemble what we saw in that atmospheric air. Okay, so then that blood is going to travel through the heart and it's going to go out through the aorta to the tissues. So now we're looking at our internal respiration. So the blood, for internal respiration is going to look just like this. So we can copy this number here. All right. So we have a PO2 of 105. And we have a PCO2 of 40. Okay, and so in the tissues are going to have carbon dioxide that they need to get rid of, and they're going to need oxygen. All right, and so you may already be thinking ahead a little. As the blood passes by these tissues, it's going to be a similar thing that'll happen to what we just saw. So these partial pressures will be exchanged for the partial pressures in the tissues. And so then this partial pressure here that we end with will look a lot like the partial pressure in the tissues. Okay? And so we already kind of know that this is what our deoxygenated blood looks like. All right? So in the tissues, the PO2 is equal to around 40 millimeters of mercury. All right, so those tissue cells need oxygen, and they have CO2, that's the waste product of cellular respiration, that they need to get rid of. Okay, and so we have 45 millimeters of mercury for that CO2. Okay, so then the oxygen is going to move into the tissues, right, from high to low pressure, and the CO2 is going to move into the blood from high to low pressure. All right, so then we'll end with a PO2 of 40 millimeters of mercury and a PCO2 of 45. Okay, so just like we saw in the lungs, as that blood moves by the tissues, it kind of takes on the pressures that we see in the tissues, and so that's our deoxygenated blood after. It then goes to the lungs to pick up more oxygen and get rid of that CO2. Okay? All right, so that's kind of what the process of external and internal respiration looks like. So now let's talk about how oxygen is transported in the blood. Okay. So oxygen is transported in two possible ways. The majority of oxygen is going to be transported attached to hemoglobin, and we know that's called oxyhemoglobin. And then 3% of the oxygen is going to be just dissolved in the plasma. And so remember, oxygen is a nonpolar molecule, so it doesn't dissolve in watery liquids very easily. So only a very tiny little bit of it will dissolve in the plasma. Okay, and so to depict the oxygen transport in the blood and visualize it, we use this oxygen saturation curve. Okay, and so this can be very intimidating for a lot of students. If you just look at it in your textbook, it might be kind of confusing, and so I've tried to break it down here for you so we can understand what it's telling us, and obviously understand what you need to know about this on your exam. All right, that's what you guys are especially interested in, I'm sure. Okay, so before we do that, let's talk about hemoglobin one more time. So hemoglobin has three major components. We have the globin part, all right, that's these blue and kind of teal chains, right? That's the protein. So it's made of amino acids. And then we have these yellow discs here. Those are the hemes, okay? The heme gives it its pigment. And then within each of the yellow discs, and I saw this, I see it all the time actually on exams, right? The iron is contained within the heme molecule, right? And it is specifically the iron that binds to oxygen, right? So iron on hemoglobin is what binds oxygen. And so every hemoglobin molecule has four iron molecules. So each hemoglobin can bind 4 oxygen molecules. Okay, so that's just a review of our hemoglobin. So then for this chart, the way I've kind of laid it out is that we have on our y-axis the percent saturation of oxygen. So this just means how much oxygen is the blood carrying at a particular time. Okay, and so on the bottom we have the partial pressure of oxygen. All right, and so this is just how the blood responds to changes in partial pressure, which is kind of what we were looking at in that picture before, right? So here, so as the blood approaches, in this case, right, a lower partial pressure, what is it going to do, right? Is it going to give up oxygen or not? Okay, and so that's what we'll look at here. And so to kind of visualize what's going on, what we can do, I've drawn these hemoglobin molecules, all right? And so each of these would be an iron. And so when it's red, that means it has all 4 irons bound to oxygen. All right, so then this one would only be binding 3 oxygens. That's why we're at 75% saturation. 2 oxygens brings us to 50%, and 1 oxygen brings us to 25%. Okay, so then 0 would be here at the bottom. All right, so that's the idea of the graph. And so we can look mainly in 2 places. So we have our kind of average partial pressures that we see in the lungs, right? We said it was around 105 for oxygen. And then we have our average partial pressure here that we see in the tissues. Okay, so 40-ish is, what we saw. It can be a little lower than that depending on how in need of oxygen the tissues are. Okay, and so what you can see in the alveoli, right, in the lungs, the saturation is typically around 100%. That makes sense because when the blood goes by the lungs, it's going to be picking up oxygen. So it'll be around 100%. And so if we follow this up, right, so from here, I just drew this line here so we can look at it in the lungs, right? We follow it up to our actual curve, which is this black curve here, all right? And then we follow that over to get our percent saturation. It is very close to 100%. Okay, so our hemoglobin would have all 4 oxygens bound. All right, that's as they pass through the lungs. And then in the tissues, where the tissues are in need, it's interesting because, so if we look at around 40, all right, which is what we were looking at with our tissues, we can take that up, all right, and that will be right around 75%, close to 75%. If the tissues are a little more in need down here, that takes us kind of closer to 50%. So in the tissues, the oxygen saturation is around 75 to 50%, meaning when the blood goes from the lungs to the tissues, it will give up only 1 or 2 of its oxygens, right? And it'll still hold on to those other 2 or 3. So it's not giving up, as it goes by the tissues, it's not going to give up all of its oxygens. Okay, so we'd either have it here, where it's only given up 1 and it still has 3, or here, where it's given up 2 and it still has 2. Okay, and that's important because the needs of the tissues change. All right, and so that hemoglobin needs kind of a reserve of oxygen to be able to deliver to the tissues if they become even more in need than normal conditions. Okay, so now let's talk about the two parts of this curve. So this is not a math class, so we are literally going to call them the flat part of the curve. So this part, right, where it's kind of nice and flat, and we're going to call it the steep part of the curve here, where it goes down really steeply. Okay, so we're keeping it simple. All right, we have the flat part and we have the steep part. They're each important for a separate reason. Okay, so the flat part is important because the oxygen saturation is totally unaffected. It stays right around close to 100%, all right, even with pretty big changes across here in partial pressure of oxygen. And so the reason that is important is because you can change, right, atmospheric pressure depending on where you are. So changes in atmospheric pressure. So let's say you're, maybe here in Georgia, right? And you go and travel to Colorado, you're gonna go skiing or something, you're up in the mountains. That change in altitude causes a huge change in partial pressure. And so you can change that partial pressure like this, and you'll still get the same amount of oxygenation of your hemoglobin. All right, so it allows us to travel and change that atmospheric pressure without it having a huge impact on the oxygenation of our blood. Okay, so the flat part means that changes in atmospheric pressure don't have a huge impact on our percent saturation of hemoglobin. The steep part, however, all right, because it's steep, that means that very small changes in partial pressure cause big changes in our saturation. All right, and so oxygen saturation in this area is very sensitive to pressure changes in the tissues. Okay, and so this is the range of where those tissues are. All right, so as the tissues become more in need of oxygen, that hemoglobin becomes much more ready to give up oxygen to the tissues. Okay, so it's very responsive. The steep part means it's responsive to the needs of the tissues. All right, and so then in addition to the parts of the regular curve, the flat and the steep part, our curve can shift either to the right or to the left, okay? And so let's talk about each of those shifts. So if the curve shifts to the right, and so I'll kind of just draw this so we can tell, right? So the right shift would be here in purple, okay, versus a left shift would be what we're seeing here in green, all right, a green dotted line. Okay, so when a curve right shifts, that means that the oxygen is going to bind more loosely to the hemoglobin. And so I think of it as hemoglobin with a right shift becomes more generous, right? It's more willing to give up its oxygens. And so if we look at an example, if we pick 40, all right, for our tissues, if we're at 40 with a right shift, right, if we go from 40, we go up, that on our right shift would put us closer to 50, right? Let's say maybe we're at like 60% versus our regular curve, we were at 70%, okay? So it's closer now at 40 to giving up 2 of its oxygens, all right? So it becomes more generous. And so this is caused by things that signal kind of distress or intense activity in the tissues. So if there is carbon dioxide, right, increasing, building up in the tissues, that will cause this. If there's a decrease in pH, that will cause a right shift. Also, if there's temperature, because more cellular respiration produces heat, so an increase in temperature, all right, so if there's an increase in temperature, that could also shift this curve to the right. And so then your body knows you're more active, you're doing more cellular respiration. That means the blood needs to get you more oxygen. So it shifts that curve to the right. Okay, and so then the opposite of all of that is true for a left shift. So with a left shift, the oxygen binds more tightly, all right? So that hemoglobin becomes really stingy. It really doesn't want to give up its oxygen. Okay, so if we follow our 40 again up, but we go all the way up to our left shifted line here, all right? Now we are above 75% for our percent saturation, all right? So it's less likely to even give up one of its oxygens, all right, at this 40 when we are shifted to the left. And so then kind of the opposite signals would cause this. So lower CO2, increase in pH, and a decrease in temperature. Okay, so that's just signals that there's not a lot of cellular respiration going on in that particular tissue. Okay, so one interesting thing about this curve and this left shift is the effect this has on hypothermia. All right. And so with hypothermia, you know, their body gets really cold, below normal body temperature. And the reason hypothermia is so dangerous is because of this left shift. So a decrease in temperature means that this is going to shift, and that blood is not going to give up its oxygen very easily, right? So your blood here, right, can pick up oxygen just fine. It's oxygenated, well oxygenated, but it's getting to the tissues, and because your body temperature is so low, it's not giving it up when it gets to the tissues. And so essentially, with severe prolonged hypothermia, your body can starve for oxygen, and essentially your tissues suffocate, all right, because that hemoglobin is not giving up oxygen. Okay, so that's oxygen transport and the oxygen saturation curve. So you should know each of these things. You should know kind of what the flat part means, the steep part, and kind of understand the right and the left shift. Okay. Our exams don't include pictures, all right? So you won't see a picture of this graph, but we'll definitely talk about it and ask conceptual questions about it. All right, now let's talk about CO2 transport. So carbon dioxide, how is it transported? So it's transported in three possible ways. The first way, it can be bound to hemoglobin. And so bound to hemoglobin, we've learned that is called carbaminohemoglobin. That is about 27% of the way carbon dioxide is transported. A little carbon dioxide can also be dissolved in the plasma. That's about 7%. And then the remaining 30% would be carbon dioxide being converted to bicarbonate, and then that is just released into the plasma and it travels that way, so in the form of bicarbonate. And so then there's an enzyme inside of red blood cells called carbonic anhydrase that is responsible for this reaction. So it converts CO2 to bicarbonate, HCO3 minus. All right, so let's look at this and what's going on here. Okay, so if we start in our tissues here off to the left side, all right, our tissues have CO2 that they need to get rid of. So that CO2 is going to move from high to low pressure into our red blood cell. Once it's in there, that carbonic anhydrase is going to convert it into carbonic acid, H2CO3, and then a hydrogen ion and bicarbonate. Okay, and so then that bicarbonate is pumped actively, right, that requires ATP, out of the red blood cell. CO3. Oh, sorry, it's not pumped. It's exchanged. This is a cotransporter. All right, so it's exchanged for a chloride ion. So that chloride comes in. Sorry, I misspoke. That chloride comes in, and the hydrogen or the bicarbonate goes out. Okay? All right. And so one thing, if you're looking at this, you might be wondering, right, well, now we've got all these hydrogen ions inside of the red blood cell. Isn't that bad for all the protein, the hemoglobin in the red blood cell? Well, a cool thing about the hemoglobin is that it actually has the ability to buffer that hydrogen. So the amino acids on the globin part of hemoglobin can bind to and buffer the hydrogen. And so that's what happens to that hydrogen. All right, so now we have our bicarbonate. It's in the plasma, it's traveling, and then it's going to reach the lungs. And so in the lungs, we need to reverse this process. All right, so that bicarbonate is moved back into the red blood cell, and then it is converted back into CO2, all right? And that CO2 moves out to the lungs based on those differences in pressure, and from there, it would be exhaled, okay? All right, so not too bad, right? Carbon dioxide definitely easier than oxygen with the oxygen saturation curve. Just make sure you know this formula for converting carbon dioxide to bicarbonate, and then the formula, for carbonic anhydrase, or not the formula, the enzyme, carbonic anhydrase. And then this, I'll just add this in here, co-transporter. Maybe, it's not gonna fit. For our bicarbonate and chloride. You can also call it an exchanger since they're moving in opposite directions. Okay, and our last topic for this chapter is going to be control of respiration. Okay, and so control of respiration comes from the brain, all right? And then it can be, we'll talk about some influences on that. So it can be influenced by levels of different gases and that sort of thing in the blood. Okay, so let's talk about control in the brain first. So in the brain, there are breathing centers. They're located in both the medulla and the pons, all right? So both of these are structures in the brainstem. So we'll talk about the medulla first, because the medulla is kind of where breathing is initiated, and then the pons can influence that breathing, okay? So the medullary respiratory centers include two groups. So we have the dorsal respiratory group, the DRG. This is the major inspiratory center, all right? So this is the center that activates that diaphragm and the external intercostals, causing you to inhale, okay? So this DRG is going to control those muscles. And then the second part of our medullary respiratory centers is the ventral respiratory group, the VRG. And so they're going to take that activation of those muscles and they're going to set the rate, all right? So they set that normal breathing rate, that 12 to 15 breaths per minute that's normal, okay? So these two together kind of set normal breathing patterns. That's in the medulla. And then we can talk about the pontine respiratory center. Okay, so the pontine respiratory centers, there's two of them, and we're, as you can tell, not going to go into a ton of detail about them because it gets very complicated and it's actually still something that's being studied. So what I want you guys to know is that the pontine respiratory centers function to influence the rate set here by this VRG. All right, and so there's different ways they can do that. So it's the apneustic center and the pneumotaxic center. The apneustic center can act on the VRG to alter the rate. This pneumotaxic center can act on the apneustic center to alter its alteration of the VRG, and they're all kind of interconnected, but don't worry about all of that. Just understand that the rate can be influenced by these groups in the VRG, or in the pons, sorry. Okay? So we have our major breathing centers kind of setting up breathing are in the medulla, and then that can be influenced by the centers in the pons. All right, and then lastly, let's talk about some influences on breathing, all right? So these are going to be things, so receptors and things that can alter breathing. So first we have chemoreceptors, and so we have two types. We have central and we have peripheral. Our central chemoreceptors are actually going to be in the brain, and they're going to monitor CSF, okay? And so they're monitoring CSF for the levels of oxygen, carbon dioxide, and hydrogen, right? It seems like everything's monitoring those, whether it's for blood pressure or respiration. we're measuring these things because they're so critical. Okay, and so just an example, if there's an increase in carbon dioxide and hydrogen in the CSF, then these chemoreceptors will trigger that an increase in the respiratory rate needs to occur. Okay, and then we have peripheral chemoreceptors. These are the ones we're familiar with from when we talked about the heart. They're in the aortic and the carotid bodies. All right, so they're going to monitor the blood going to the head and to the systemic circulation, also for those same things, CO2, O2, and hydrogen. All right, so that's pretty straightforward. We dealt with similar receptors before. All right, we've also talked about baroreceptors before. Okay, and so these are going to be those same baroreceptors. So these baroreceptors detect stretch or pressure on blood vessels. All right, so that would essentially be blood pressure. And so respiratory rate responds to blood pressure. And so as your blood pressure increases, your respiratory rate will also increase to get more oxygen to the tissues, because that increase in pressure kind of indicates you're pushing more blood to the tissues. Okay? All right, so these mechanoreceptors now are kind of unique for the lungs, and they're very interesting. All right, so these mechanoreceptors, so mechano just means that they respond to some sort of physical change, all right, mechanical change. And so they're located in the lungs themselves, and they monitor how inflated the lungs are, all right? And they're responsible for a reflex called the Herring-Brewer reflex that helps to prevent your lungs from overinflating and kind of tearing that delicate lung tissue and damaging the lungs. So they prevent overinflation. So if you inhale too much and stretch your lungs too much, this reflex will kick in and it will just shut down inspiration, all right? That's because these mechanoreceptors have detected that the lungs are getting too inflated. All right, and then we all probably know by now that breathing is closely associated with the limbic system, right? Your emotional center of the brain. So if you're anxious or in distress, you'll start breathing really, really fast, right? You may even, if you're having an anxiety attack or something, start, hyperventilating and that sort of thing, okay? So anxiety and distress can increase a respiratory rate. And then also the cerebral cortex, right? Particularly that precentral gyrus you learned about last semester, can control breathing, because the diaphragm and your external intercostals, those are skeletal muscle. So you can voluntarily control breathing. And so the cerebral cortex can kind of interrupt those medullary centers and take over breathing, all right? Okay. And then respiration and pH are very closely related, as we saw with our chemoreceptors, right, monitoring hydrogen. pH and respiration are very interdependent. And so that's actually because as CO2 increases, the pH will go down, okay? And so then the respiratory rate will act to counteract that, right, and bring that CO2 down by increasing the rate. So there are two situations that can change that CO2, breathing-related situations, that can cause the CO2 to change and alter the pH, okay? So it's two types of breathing. So hyperventilation means that you're over-ventilating, right? You're breathing very fast and very deeply, and so you're getting rid of too much CO2, okay? So you're over-ventilating, you're getting rid of too much CO2, all right? And so that is called hypocapnia, all right? When your CO2 levels are too low, hypocapnia. So then this would elevate pH, and so we call that respiratory alkalosis. Specifically respiratory, because it's being caused by hyperventilation. Okay? So then the opposite situation, hypoventilation, this would be underventilation, all right? And so this would be like very shallow breaths, right? You're not getting rid of enough CO2, So CO2 will start to build up, leading to hypercapnia, too much CO2 in the blood, and that would make the blood very acidic. Okay, so that would be acidosis, and because it's caused by hypoventilation, it's called respiratory acidosis. Okay. All right, so that wraps up the respiratory system. And so I will talk to you guys next time.